We hope that Ajinomoto will grow up into the company that will play a great role as a leader in the next generation.
Thanks to Ajinomoto's great support, Heartseed can optimize the protocol of the differentiation and purification of cardiomyocytes.
If we can show the safety and efficacy in this clinical trial, we can obtain the manufacturing approval in Japan. We believe that Ajinomoto is an important partner in its development.
Eliminating of the residual iPS cells are critical to prevent teratoma formation, cause a teratoma formation, and it's a high hurdle to create the iPS cell therapy product. Especially our MOA mechanism of action is a wrist muscularization, so the cell needs to stay in the heart ventricular wall and stay for a long time, so that undifferentiated cell might cause a teratoma formation, so the elimination is quite important. The Ajinomoto provided sample media for many times, and Dr. Fukuda's lab test the media and give the feedback to Ajinomoto. And Ajinomoto provided the media many times, and thanks to the Ajinomoto's contribution, we finally developed the differentiation and the purification protocol. Thanks to Ajinomoto's great support, Heartseed can optimize the protocol of the differentiation and purification of cardiomyocytes. Heartseed can reproducibly manufacture cardiomyocyte products. Our mechanism of action is a remuscularization that no precedence exists. In that situation, we have to develop our way by ourselves. So we had many discussions with PMDA before the clinical trial notification submission. There are three key success factors to achieve the speedy approval of the clinical trial notification. The first, our preclinical data is very clear in the safety and efficacy. So based on the very clean data, we discussed the PMDA rapidly and get a very positive feedback about the clinical notification. And secondly, PMDA reviewed all raw materials and of our product. Of course, including the media is a key discussion point. Ajinomoto provided support to answer the question of the PMDA. Thanks to the Ajinomoto's great contribution, the PMDA agreed our manufacturing process and start the review of the clinical trial notification. And finally, our research collaborator, Keio University, have a deep insight of the cardiomyocyte manufacturing, and they are supporting us to provide the answer to the PMDA's question. Yeah, obviously the culture medium is very important for iPS, for manufacturing of iPS cell-derived product. To reach out to the worldwide market, we need to manufacture the more and more amount of the iPS cell-derived product. In that situation, the quality assurance and cost reduction of the media is a key critical factor. I appreciate the close collaboration across the Ajinomoto's department in charge of the business development, the manufacturing, and research and development. I hope that the Ajinomoto will continuously play a vital role of the manufacturing the media.
I started myocardial regeneration research using stem cells in 1995. Initially, we demonstrated that cardiomyocyte can be regenerated using bone marrow mesenchymal stem cells. Since then, a large number of cardiomyocytes were required for the treatment of heart disease. We have shifted our research to human ES cells and iPS cells. The research has progressed, and human cardiomyocyte can be obtained from these cells. However, it has become clear that the residual undifferentiated iPS cells and a mixture of non-cardiomyocytes are the problems for the clinical use of myocardial cells produced from iPS cells. In the process of purifying cardiomyocyte, we found that cardiomyocyte and iPS cells have different energy metabolism enzymes and require different nutrients for the cell survival. Therefore, I came up with the idea of purifying cardiomyocyte using the ingenious composition of the cell culture medium. When I reported it at the academic conference, a researcher of Ajinomoto, Mr. Okamoto, listened to my talk and had a discussion after the lecture. From that time, our collaboration between Keio University and Ajinomoto had started. By putting various ideas into the production of a cell culture medium, I found that there were endless possibilities of the cell culture method. So I decided to proceed my research together with Ajinomoto. After that, I founded academia-based bioventure Heartseed in 2015 because of the prospect that myocardial regeneration therapy will be realized. And now, I'm currently conducting clinical trials. We believe that Ajinomoto is an important partner in its development. I thought that each composition of the cell culture medium have a special meaning. There was optimum concentration according to its purpose. For this reason, I plan to jointly develop a cell culture medium with Ajinomoto. One of the great things about Ajinomoto is that it has a high ability to analyze the composition of the cell culture medium. For example, regarding how the components of the cell culture medium changed before and after the cell culture. The latest science, such as mass spectrometry, is used to analyze how each component changed. This was an extremely effective in finding the optimum condition of the cell culture. Secondly, according to our requirement, we asked Ajinomoto to adjust each component of the cell culture medium and to engage in a huge amount of experiment to determine the optimum concentration of its component, and experimental media with various compositions. This made it possible to produce a cell culture medium with favorite conditions that cannot be achieved with existing media. As a result, we were able to obtain 3 culture media: iPS cell amplification medium, cardiomyocyte differentiation induction medium, and the cardiomyocyte purification medium. I think that Ajinomoto's high level of science and the breadth of the bosom that did not rush the results were great. Regenerative medicine is expected to become an important medical treatment in the 21st century. To date, most of the researchers and companies purchased the common existing cell culture medium. However, in the future, each cultured cell have optimum cell conditions. So it will be necessary to use the best cell culture medium for each cell. Ajinomoto has the ability to analyze the composition of the cell culture medium and metabolic pathway of each cell in detail. By fully utilizing this power and supplying cell culture medium to this new industry area, we hope that Ajinomoto will grow up into the company that will play a great role as a leader in the next generation. Heartseed is currently conducting a clinical trial of human iPS cell-derived regenerated cardiomyocyte transplantation into the patient with congestive heart failure caused by ischemic heart disease. If we can show the safety and efficacy in this clinical trial, we can obtain the manufacturing approval in Japan. In addition to the myocardial regeneration medicine, we would like to develop regenerative medicine technology in various fields using our iPSC technology.